Hi everyone, I wanted to record a presentation for you to go into more detail on the pharmacogenetic aspect of pharmacology. So this is really digging in deeper to chapter 10, which really delves into the genes and the drug metabolism. And so basically what we're talking about is how inherited differences, so how genes and genetics can impact therapeutics. Also, we are going to gain an insight into how genetic information can really predict how an individual is going to respond to a drug. So to understand our terms, pharmacogenetics refers to a single gene or a few gene variations, and we call those polymorphisms. Pharmacogenomics is kind of a broader term. It's more genome-wide. However, you kind of hear these used interchangeably, which is just becoming part of the lingo of people, but this is kind of the true form for you. Okay, we have to kind of start with some basics. I know you're all familiar with this. This is the human genome. This shows our 23 chromosomes that we've reviewed, and so we're going to just kind of dig a little deeper into some genetic concepts. We all have our own unique DNA found in our genes. Identical twins share their DNA. And what you need to know is that DNA is packaged and kind of compressed and wound down into a chromosome. We've got 23 pairs in humans. Every chromosome can have anywhere from hundreds to thousands of genes. We've got more than 25,000 genes on the human genome. And this is really where what makes us human, how our proteins work, this is kind of where it all takes place. Now, as you can see here, you've started at the bottom there with the chromosome. It is unwound, and remember, histones are like those little discs that the genetics are wound tightly around. And when they get all the way unwound, you see the actual DNA. And those are macromolecules that bind together, and a segment of this DNA is a gene. Here's a picture that actually shows a segment. Now later we're going to talk about there are certain areas that actually define the segment of a certain gene, which is important when transcribing a gene. What is the big deal about genes? Well, they're the blueprint for all of the protein. Human functioning and the type of proteins totally is dependent on the blueprint and the genes. So we've got 3 billion pairs, base pairs in the human genome. Here's a picture of the base pair. You can see C and G, which is cytosine and guanine, and they always match up together. And then you've got T and A, which is thymine and adenine, and those are always going to be paired together. So these are macromolecules or nucleic acids. They bond together via hydrogen, and they become what we call base pairs. Now, their function is to store and express our genetic information. There are 3 billion of these in the human genome, and what's fascinating is that 99.9% of them are the same among all humans. So there's only 0.1% for variation among individuals. But think about how much variation there is. When you've got 3 billion, 0.1% is, a fair amount. Okay. In our 23 pairs, we inherit one of each of the pairs from each of our parents. And this is what, determines our sex, our color of our eyes. But in this presentation, we're wanting to know about what determines how we respond to drugs. Okay, allele is an important concept. It's a version of a gene. So you've got your chromosome, you've got your, we'll just say 2,000 genes on a particular chromosome. Each gene has a certain location or version, and that's an allele. So The capital letter is commonly the dominant version, or if that's what you inherit, that's what's going to be expressed. The small a is the recessive version, and then at the bottom you can see kind of what happens when you get, when the genes get scrambled and the DNA and the chromosomes are made, which version you get. If you're homozygous, the two alleles are the same. So the thing to remember here is that an allele is a version. So how your patient responds to drugs will depend on which version or which allele of a certain enzyme that they received in the gene pool. The whole big purpose, the whole big shebang of all this is the genetic code. Now, as you know, the genetic code, that's the instructions for making a specific protein. And proteins, as you know, are made up of amino acids, and those are on the right. The sequence gets transcribed. So the purpose of this slide is to kind of show you the transcribing aspect of it. And this is where polymorphisms or difference can kind of come into the picture. So sometimes we get variations in these base pairings, and that's what leads to differences. It's going to get really exciting now because we're going to really talk about those individual variations with the same drug. So you line up 20 people and you're giving each of them a dose of, let's just say, citalopram, Celexa. You are running a mental health clinic, and many of your patients that you're seeing today are depressed. What we're going to look at here is how they're going to respond. In the middle, what I want you to look at in this graph is the middle, the grayed area, that's the therapeutic range. That's where you're aiming. Those pharmacokinetic studies in the drug studies that we talked about, this is what they came up with. We want our drug to be here. In other words, this is where we're going to get the best effect with minimal toxicity. So that's the green line. The bottom line, those are the ultra-rapid metabolizers. As you can see, they really barely get to the therapeutic range, okay? So they're going to metabolize it quickly, and then it's going to be excreted. At the top, you have the slow metabolizer, and that is your person who's at risk for toxicity. So in the example of the citalopram, those patients are not going to be able to break it down, and they're going to tell you, oh my gosh, I felt sluggish, or I was terribly nauseated and I just stopped taking it. This is the individual variation that, as you will see, is due to genetics or pharmacogenetics. Okay, there is a whole spectrum of variation, and we're going to look at that next. So you can have a poor metabolizer, which is going to be homozygote for decreased expression of the particular enzyme needed to break it down. Then we have the intermediate metabolizer, so they're going to have a heterozygote, but they're still going to have some variation. Next, you have the normal metabolizer. Now, of note, there are two kind of phrases that you're going to see which can be misleading. This extensive metabolizer is synonymous with normal. It's just, the nomenclature that we have, we've got poor, intermediate, extensive, and then we move into ultra metabolizer. So extensive is normal, wild type is normal. Don't ask me why. The rapid metabolizer, they've got many copies of the gene. They also can have a variant in the promoter gene, which we're going to talk about. The ultra-rapid metabolizer, they've got multiple copies, and they're even more quickly going to metabolize the drug. And your patient's going to say, well, that drug absolutely did nothing. And if you think back to our graph on the earlier page, they're the ones that barely made it into the therapeutic range, okay? Okay. Now this pie chart didn't include the intermediate metabolizers of the previous slide, but it kind of shows you the percentage of the population that has each one. Now, as they're explaining it in the bottom of the key, they have this *1, *1. So that is kind of geneticist speak for locating alleles. Remember, allele is a version of the gene, and so they've kind of adopted using the star to describe the location, okay? The point of this slide is that when we have genetic testing available to us, we can predict and we can know which type of metabolizer the patient is because we get a preview into their genetic sequencing. Okay, now we're going to talk about polymorphism. Another word for polymorphism is variation or diversity, and it can be everything from a single base pair substitution. So at the top you see this individual had a T or a thymine, and then when it got copied, an adenine is substituted. Or down at the bottom, it can be several repeats. Okay, so what you can tell is that I just received an email. That's what that little bling is, but we're not going to look at that email because we're doing this. Okay. Polymorphism, as I said, is diversity. It's the genome of a species. It's the place in the DNA sequence where there's variation, and it's indicated by a star. In order to be a polymorphism, it has to occur in at least 1 in 100 people. It has to be at least 1%. Haplotype is another word that means there's more than more than 2 variants that are inherited together. It's not a mutation. A mutation is more on an individual level that typically leads to an abnormality. Here we're simply talking about diversity, variation, and specifically the genes responsible for drug activity have different alleles on the same gene, producing different metabolic effects. The type of metabolizer and the drug. Now, in class, we spoke about the prodrug and the active drug. As you remember, prodrug is administered or ingested in its inactive form, and it has got to be converted to become active. It needs a certain family, a certain enzyme. So your poor metabolizer, in other words, they don't really have that enzyme, or they are homozygous for the one that doesn't really metabolize, they're not going to get a therapeutic effect. An example for you, clopidogrel or Plavix has got to have CYP2C19. Codeine has to have CYP2D6. Both of those, they're prodrugs that will not be therapeutic if they don't have the particular enzyme. Conversely, if they are rapid metabolizer, that will be a different effect, which we'll talk about. Active drug is already active. It does not have to be converted into anything. What the active drug has to have is it has to be metabolized. It has to be broken down and excreted by certain enzyme. So if you have a poor metabolizer, that drug is going to build up because they're not breaking it down. For example, warfarin needs CYP2C9 in order to be eliminated. So in CYP2C9, the 2 main polymorphisms are the star allele, *1 and *2. And remember, star allele stands for variant loss of function allele. It's a location. So *1, they've got normal. *2, they have a 30% decrease. *3, they can have as much as a 90% decrease. So your patient, if they have a *3 allele, probably clopidogrel is not going to get metabolized to therapeutic effect. So this is something that is nomenclature in genetic testing. Now there's other things that can alter metabolism. Remember we talked about the inducer and the inhibitor. So the inducer accelerates enzyme activity. It can be a drug or a chemical. It could increase the expression of the enzyme. For example, phenobarb is a drug. So a drug induces the activity of CYP3A4, which means it's going to be increasing metabolism. So the rate of metabolism will be faster if you take phenobarb along with, let's just say, amlodipine. I'm not sure amlodipine needs 3A4. It's just an example I'm using right now. But if you take phenobarb and 3A4, phenobarb is going to induce the activity of that amlodipine, and it's going to get quickly metabolized. So it accelerates the activity, so the amlodipine is going to get pushed out into the system. It's going to quickly lower the blood pressure, but then it's going to wear off. Your patient might complain, wow, that drug really kind of made me dizzy. And if they had checked their blood pressure, it might be something like 90/60. And then they'll say, but 2 hours later, I had a headache and I checked my blood pressure and it was 170. So things to think about. Inhibitors slow down the activity of that enzyme. Grapefruit juice slows down or inhibits 3A4. So a 3A4 substrate will be more slowly metabolized. The point is, genetic variation can affect the pharmacokinetics and also the pharmacodynamics. This shows our famous pharmacokinetics, our ADME, our pharmacodynamics, what the drug does to the body, which can be receptors, ion channels, or enzymes. Remember we spoke about promoter genes earlier? That's an area on the DNA that turns the gene on and off. And actually, the really cool thing about a promoter gene is it's where the messenger RNA is going to bind. And so if your promoter gene, if you've got a polymorphism that turns it off, the RNA is not going to bind there, and so that gene is not going to get transcribed. Now, the CYP450 system and pharmacogenetics. As you know, cytochrome P450 is a metabolizing system. It's an enzyme system dedicated to metabolizing drugs. It's in the liver, it's in the small intestine, it's in other places, but it's unique in each person. So about 90% of drugs that have to undergo phase 1 biotransformation, they're substrates of a particular P450 enzymes. Our genetic polymorphism is differences in this enzyme system, and it can be individual, ethnic, it can be like Korean, Asian, African American, Caucasian, and there are also gender differences in these systems. The bottom line, it explains why individuals differ in their response to the same drug. We're mostly concerned with 6 primary enzymes, which 75% of all drugs need. The first 3, 3A4, 2D6, 2C19, you see the percentage of drugs that need a CYP450. That's the enzyme that they have to have. The little hashtag, or the little tent, that is signifying an enzyme that has a lot of genetic variability. And here are 3 more. Together, these 3 are 15%. So 2D6, 2C9, 2C19 have a lot of genetic variability. Now, remember that substrate is a substance, typically a drug, that needs a certain isoenzyme. It has to connect with that isoenzyme in order to be metabolized. Codeine is a substrate of 2D6. Codeine is taken as a prodrug, as we said earlier, has to have 2D6 to become active. Fluoxetine is not a prodrug, but it needs 2D6 to be metabolized. So it's 2 different things, and I really want you to think about this, okay? So the ultrametabolizer who takes codeine, remember codeine's a prodrug, so it's an ultrametabolizer, so their 2D6 is going to be really fast, really efficient at metabolizing the codeine into the active morphine. This is going to shift their AUC, their area under the curve, from kind of a sloping to they're going to have a high peak and then a quick drop off. They're going to tell you, oh my gosh, codeine snows me, but in a couple of hours my pain comes back. I hate that stuff. Whereas a poor metabolizer, so they've got the polymorphism where they don't have many of the 2D6 enzymes, so they're going to poorly metabolize it. They're not going to have the oomph to convert that to an active metabolite. You're going to hear, codeine didn't do a thing for me. Now really think about that. Really kind of understand why each one happened. Now, Strattera was the first drug to come out with a pharmacogenomic warning because it needs 2D6. They said that in a poor metabolizer, decreased appetite and insomnia occurred twice as often as with an extensive metabolizer. Now, Strattera is not taken as a prodrug, so the poor metabolizer can't quite clear it from their body, so they're going to have more side effects. We're going to talk about CYP2C19. These are its substrates. A slow metabolizer is going to get a therapeutic effect with about half the dose of normal metabolizer, except for clopidogrel, which is a prodrug. So it's kind of different, but you can screen for the 2C19 variants. These are the things that we know can be mutations on the 2C19 gene. It can be wild type. Remember, that means normal. It can be star 2, which is the most common, and it's a codominant trait, mostly in Asian. Star 3 is less than the population, and that's also Asian. Now, an exon is another important concept. An exon is that part of the gene that codes for the amino acids. So most gene sequences are kind of broken up. So the exon codes for a specific amino acid. Remember, a gene is a whole sequence for a protein, and proteins are made of specific amino acids. Okay? The CYP2C19 has 9 exons, in other words, 9 areas for amino acids. It's on chromosome 10, and we've found more than 30 polymorphisms. Remember, clopidogrel Plavix, we take that for our patient who has a stent, a fresh stent, and we want to inhibit platelet activity congregating around that stent, blocking it off. The MOA, there you have the mechanism of action of clopidogrel. So clopidogrel has to get, it's a prodrug, it has to get modified to the active form, about 15%, and then 85% is hydrolyzed and excreted. So successful treatment is critical to metabolize this properly. So an inherited polymorphism can really impact your therapeutic effect. You can have normal, you can have reduced, or you can have absent enzyme activity. So this slide kind of shows what can happen among the poor, the extensive, and the intermediate metabolizers. Just kind of ponder that for a moment, if you will. So in summary, in our whole unit, there are many things that impact therapeutic outcomes. We're considering pharmacokinetics, pharmacodynamics, age, lifespan in our unit. In this presentation, we are specifically looking at pharmacogenetics, how they can affect therapeutic effects, the drug interactions that are possible, as well as the inducers, the inhibitors, and polymorphisms. So these are all important concepts. I thank you for your time and attention. Here are my references. I look forward to your feedback, and also there will be a couple of questions posted to help you kind of process this and store this information into your neural pathways so that you will successfully complete quizzes and tests and be great prescribers. Take care.